Hi everybody. So this is going to be our last ed puzzle before the last test. Um, After this, we're going to be rolling over into the next try, and I will be seeing most of you guys again. For this, we're going to look at transportation in and out of the cell. In general, looking at movement into and out of it, there's a number of things that a cell needs that it can't make itself. For instance, in animal cells, we can't make our own food supply. We can't make certain ions, we can't make certain proteins, so we need to bring those things into our cells. And this is why we can't have a cell membrane that is completely sealed. It has to be that semi-permeable type of material to allow stuff in and out. So most things move in and out of the cell based off of what is called a concentration gradient. So setting aside the idea of selective permeability, the actual movement is based on this idea of a concentration gradient. And this is a difference between high concentration and low concentration. So we have an area that is very highly concentrated. There's a lot of stuff there versus an area where there's not as much stuff. So over time, that stuff will tend to flow to where there's less stuff. And this is the idea of a concentration gradient, going from high to low. And if you remember in class, to describe this idea of concentration, I gave you guys the example of the Kool-Aid. If we have a pitcher of Kool-Aid that we make according to the directions versus one that we make with half as much water, the one that we make with less water will taste stronger, which is an indicator of higher concentration. So, as you can see from the top diagram, eventually, things will start to kind of even out. And going from a spot of high concentration to a spot of low concentration, as that shift happens, they will eventually reach this spot where they become relatively equal, which is what we call equilibrium. Once they hit the spot, there's still going to be movement, so concentration might shift up one way, then it might shift up the other, but it's just going to be this back and forth. There's not going to be any real net movement in or out of the cell. There'll still be movement, just not a significant amount one way or the other. And with net movement, like I said, there's going to be movement in both directions. It's just there's going to be more movement in one way. So that will be the way that stuff tends to move, is in that direction until they reach equilibrium. And there are two types of transport. Passive transport, which is moving with the concentration gradient, going down that hill, and active transport, which moves against it and goes up the hill. The difference is that passive transport does not use energy, whereas active transport does. So, starting with passive transport. As said before, this is movement that is from areas of high concentration to low. This type of movement does not require energy. And in class, I gave you guys the example of if you're standing at the top of a hill with a ball, and you push that ball to get it rolling. Once it gets rolling, you don't have to do anything. It just does it. You don't have to put energy in to do it, to get it to move. And there are three types of passive transport that we'll look at. First is what we call simple diffusion. Simple diffusion is just the general movement of particles. And this isn't even across a membrane. I'm going to be talking about it across a membrane because we're talking about cells. But simple diffusion is the particles don't need any help at all to move. They just move. They can cross the membrane on their own. They don't need any special proteins or anything like that. And they still move from high concentration to low, as you can see in our diagram here. The blue dots move through the membrane, going from where there's a lot of them to where there's not a lot of them. The next type is what is called facilitated diffusion. So if something is being facilitated, it means it's being helped. And this is movement across the membrane with the help of some of the proteins that are embedded in the membrane. And this is used for kind of two purposes. One is to get any of the larger substances, and two, to get any of the charged substances across. So anything that can't normally just cross through the phospholipids will come through a protein of some sort. And there are two types of this: channels and carriers. So channels, think of them as like an open hallway. Anything that can fit through it can go through. Whereas a carrier is kind of like a one-way door. Once the right thing goes through, it shuts and pushes it into the cell. These are still types of diffusion. They don't use energy. They go from high concentration to low. Okay? And our third type, which we'll talk about a little bit more on the next slide, is osmosis. Osmosis is a specific type of diffusion. As opposed to the stuff that's mixed in moving, this is the diffusion of water. So the water is doing the moving as opposed to the sugar or the salt or whatever else. And this is specifically crossing a membrane. So osmosis, as I said, is specifically the movement of water from areas of high concentration to low concentration. And let me be specific, this is of water concentration. So this is actually going to be kind of reversed from what we're thinking. As opposed to thinking of that drink mix, we can think of it as the liquid part. Where there's higher amounts of water, that's going to be the areas that is our high concentration. Where there's not as much water, so where there's more mix, that's where the water is going to be moving to. And we tend to base this from the concentration of the solution outside of the cell. Because if you remember the ideas of homeostasis, the cells themselves aren't going to be changing that much. They're going to want to stay relatively stable. And that means the amount of salt, the amount of sugar, and whatnot inside of them. So their mixes are going to be relatively stable. But the solutions, the outside environment that they're in, can differ greatly. And that's where we come to this idea of tonicity. Tonicity is the relative concentration of a solution outside of a cell compared to that cell. And there are three of them that we'll talk about on the next page: isotonic, hypotonic, and hypertonic. And I talked about these in class. I want to go through all three of these. So to start with, remember, tonicity is the relative concentration of the solution that is outside of the cell. Since the concentrations inside are relatively stable, there's not going to be too much change from cell to cell. But the stuff outside can change greatly. Our first type is what is called isotonic. Iso meaning same, tonic referring to a mixture or a solution. This is when the concentration outside of the cell is roughly the same as the inside of the cell. It is at equilibrium. There is no net movement in one direction or the other. So, as we can see from the diagram, the amount of water going in is the same as the amount of water going out. The cells will stay in their normal, healthy shape. Second is what is called hypotonic. So I gave the example of a hypodermic needle. These are needles that are used to puncture and go under the skin, the dermis. With hypotonic solutions, the outside concentration, so the solute, the mixture concentration outside the cell, is lower than it is inside. Whereas the water concentration outside is higher than it is inside the cell. This will cause a net movement of water into the cell, which causes it to swell like, imagine you're blowing up a balloon and you keep blowing and blowing and blowing and blowing until pop. That's what happens with the cells as well. If they take in too much water, they will eventually what is called lyse or burst. The third type is what we call hypertonic. And for the example for this, I talked about somebody being hyperactive. They are overly active or overly energetic. Hyper means over or above. Hypertonic solutions, the solute concentration, so the mixture outside of the cell, is higher than it is inside of the cell. The water concentration outside is lower than inside the cell. So because the water concentration is lower outside compared to inside, there's going to be a net movement of water out of the cell, which, if we look, we can see it appears to be more water flowing out. And this causes something that is called crenation, or shriveling of the cells. So using that analogy of a balloon, imagine you stick the balloon in your mouth and you suck all the air in and the balloon collapses on itself. That's what happens with the cells as well. So that's osmosis. I have 2 questions that are going to be popping up here in a minute. So I will give you a second. Okay. The last thing I want to talk about is what is called active transport. So all the stuff that we've talked about up till now is passive transport. Passive transport is movement with the concentration gradient. Active transport is movement against the concentration gradient. So when we talk about diffusion, diffusion is moving down the hill from top to bottom. Active transport, on the other hand, is if we take that ball analogy, once we have the ball at the bottom of the hill, well, how do we get it back to the top? Well, we have to either pick it up and carry it, or push it and push it and push it. Either way, we have to put an effort to move from the bottom of the hill to the top, which is kind of the same idea with active transport. The main difference between active and passive transport is that active transport requires energy. It requires an input of energy to happen. And we actually see 2 different types of active transport. First is with small molecules. So these are going to be things, the main thing that you will see is if you move on to higher levels of biology, you will talk about a type of protein called a pump. Specifically, in our bodies, it is called a sodium-potassium pump. So these pumps, what they do is they pump ions into or out of the cell. But specifically, they are pumping them against the concentration. So imagine an electric pump that is blowing up, well, a balloon. It's forcing them in there. So it has to put energy into it. The second type is for bulk movements, and this is movements using vacuoles. And this can either be into or out of the cell. The first is endocytosis. Endo, meaning in, cytosis, referring to the cytoplasm of the cell. This is used to take in either extremely large or bulk materials. What happens is the cell membrane actually folds up around these to create a vacuole, pinches off, and it floats into the cell. This is a way of taking, like I said, a lot of material or very large things that even can't get through proteins on the membrane. And we also see the opposite happening, where we have these vacuoles go up and attach themselves to the cell membrane, and they spit it out. This is called exocytosis. Exit. So that's all I have for you guys. Make sure that we study for the test, and I will see you guys on Zoom when next I see you. All right. Goodbye.